Well, welcome everyone. On the kind of Zoom format I've only got a strip of about the first 5 people. So apologies if there are people that are very familiar to me on the call that I haven't said hi to, but I guess I will catch your name later when we have a discussion. Now, the slides I've prepared are really an entry level to this subject, because I thought that made sense for, potentially a very mixed audience, some of whom are new to this whole area and others that are old hands. So, if there are detailed and specific questions about, really up-to-date aspects of research in this area, hopefully that will come out of the discussion at the end. But for those that don't know me, I'm a neurologist and a researcher. I'm based in London at the National Prion Clinic, which is an NHS clinic, very closely affiliated with the Institute of Prion Diseases at UCL. And I've been working on this subject for longer than I care to think of, but I reckon about 23 years now. So when we were first born and we were cute babies in the pram, probably some of the first conversations we heard were on the subjects of genetics. That was some relative peering over the side of the cot saying that we got grandmother's nose or our father's eyes. And I think when people just generally think about genetics, one of the most striking examples are identical twins that I show here. But of course, genetics isn't just superficial appearances. It goes much deeper than that, because genes determine which diseases we're likely to get in life. And that can mean the possession of genetic changes that are extremely rare in the population and determine for sure that something's going to happen, but also changes that define differences in the entire population, common changes that are very variable between one person and the next, that determine things like your height, your intelligence, your and various other traits. So diseases are a mixture of these things. Some of them are really determined by individual genetic changes. Others are a mixture of things, are a mixture of a collection of genetic factors, environmental or lifestyle factors, and just bad luck. So, the first bot of my talk kind of introduces how those factors are relevant to CJD or prion diseases. So we see three major different types when it comes to the cause of prion disease. So the most common is sporadic CJD, and the main explanation for that, based on our current understanding, is bad luck. There's no obvious risk factor, and there's no really strong genetic factors. The subject of most of this talk is inherited prion disease, or genetic CJD, where in this case, the disease is determined by a genetic mistake, a spelling mistake in the gene code that tells the body how to make the prion protein. And I'm going to talk a bit more about that. What I'm not going to talk about is what makes prion diseases special, which is the fact that there are certain types that can be acquired or transmitted between people or from animals to people, and in the laboratory or in the wild, from animals to animals and animals to cells. They can, they behave like infectious diseases. For humans, there are certain medical procedures that are risks, and of course everyone's heard of BSE and the fact that transmitted to humans variant CJD. From the point of view of inherited prion disease, it's important to know that there are no risks for an individual to spread the disease by everyday contacts, including sex. The risks are with medical and surgical procedures, and the request is that people that have a prion disease mutation or are at risk do let their doctors know about that prior to any invasive procedures. Patients shouldn't be blood or organ donors. But let me talk a bit about sporadic CJD first, because I'm going to contrast it with inherited prion disease. So sporadic CJD is typically a rapidly progressive dementia or problem with thinking skills, and includes neurological features like jerky muscle movements and a loss of balance. Now, this is a very fast disease, tragically, and most patients will be dead within 6 months of the disease starting. It seems to occur at random. That's what I mean by bad luck. There's no obvious explanation in a person other than age. And the risk of getting this in one's lifetime is about 1 in 5,000. And that's what leads cumulatively to the commonly spoken about figure of 1 or 2 per million per year. It doesn't run in families. Of course, eventually you will find a family where through 1 in 5,000 there are 2 people that get it in the same family, but generally it doesn't run in families. So we think the cause of sporadic CJD is some spontaneous microscopic change in a protein shape that happened at one point in time. It may also be triggered by a gene mutation in an individual cell, but not a gene mutation that can be passed on in families, just something that affects an individual cell at one point in time. And we very actively research sporadic CJD to try and understand genetic risk factors that may modify these risks. But these are small effects that we're looking to detect. We're looking to try and understand sporadic CJD better so that we might be able to tackle it, because being able to tackle a disease comes from understanding it thoroughly. So if I talk about genetic risk factors for sporadic CJD, I'm talking about something like changing your lifetime risks from 1 in 5,000 to 1 in 4,000. It's not really relevant information for an individual. But the inherited prion diseases are different. And I'm going to go on to explain why. But first, I just want to explain a bit about what the symptoms are like, because they're extremely varied. They only comprise about 10% of new patients we see, the vast majority of sporadic CJD. And they can look like sporadic CJD. So patients with inherited prion disease can look just like CJD with a very fast disorder affecting thinking skills and neurological features. But there are many different types that we recognize. For example, some present with fragmented sleep problems, labile blood pressure, changes in temperature and sweating that we refer to as fatal familial insomnia. And there are certain types that cause balance problems first and go much more slowly than CJD, in that they last for perhaps 4 or 5 years in total. There are other types we've seen that really just affect thinking for a very long time, maybe a decade or more. And very rarely we've seen families where there's actually a non-neurological presentation with diarrhea and sensory changes prior to any problems with thinking skills. So quite a diverse set of different symptoms that are largely determined by the exact spelling mistake or genetic mistake that is in the prion protein gene. So when we go to see patients that are suspected to have prion disease, we do recommend a test to all patients with apparently sporadic CJD, because occasionally we find gene mutations are the explanation, even if there is no signs of that running in the family. And if we find one of those errors, this is the explanation for the disease. It's a very strong risk factor that determines whether someone will get the disease in their lifetime. So when we do research in this area, when we research inherited prion diseases, we're interested in different questions other than finding the cause. We try and find out information about what type of disease a gene mutation causes, develop certainty that that gene mutation causes the disease, and we are interested in researching ways to better predict when symptoms might start if somebody is known to be a gene mutation carrier. So there are some special opportunities as well in inherited prion disease. You can decide to have a test to see if you have inherited a gene mutation that is in the family. Some people may decide whether or not they might want to have children that could inherit the mutation, or use technologies that could make sure if you do have children that they don't carry the mutation. And there are also opportunities for research, either to join studies that are looking at people over time to try and diagnose when the symptoms might start with more accuracy, or even interventions like treatments that aim to prevent the disease starting at all. So in terms of clinical diagnosis, the usual tests for sporadic CJD are not so useful. Brain scans may show signs. They may alternatively just show some shrinkage of the brain that isn't specific to the disease. Brainwave tests are often just show slowness rather than the specific features of CJD, and spinal fluid tests are often normal. It's the gene test in this case that is definitive. So everybody that has inherited prion disease has a mutation or a spelling mistake in the code that makes the prion protein. There are no exceptions to that. So I want to talk a bit about molecules for a while, because you do have to, you do have to understand the molecular processes thoroughly to, to have a complete understanding of what's going on. So in order to do that, you need to know a little bit about how our bodies are made up. So our bodies are comprised of cells. These, blood cells are a good example, but there are skin cells, muscle cells, brain cells, and they adapt to do all the different functions that our body needs. The cell contains a nucleus, typically in its center, and the nucleus contains all wrapped up the DNA code. And the DNA code of the cell tells the cell how to make its proteins, when to make them, and how strongly to make them. And this code comprises 4 different letters and is 3 billion letters long, and it's all wrapped up like a telephone cable in a tiny little part of the cell. So the DNA code is packaged up in the nucleus. Little signals are sent out, little messages are sent out of a related compound called RNA, and then the RNA is translated to make proteins. So this is how it works. Proteins are made up of amino acids. There are 20 different amino acids. Now I mentioned there are 4 letters in the DNA or the RNA code. So you need multiple letters of that code to tell the body which amino acid to make in the protein. So this is done inside the cell, and the machinery looks at 3 letters of the code at any one time to select which amino acid is made. And here you can see the amino acid, the chemical, is represented by a little yellow dot. And this is the growing protein that has been determined by these colored codes on the RNA. And these blobs here represent the machines, the protein machines that control this process. So this is how the prion protein is made from the code for the prion protein in the gene. So look at a nerve cell. This is where the prion protein sits. So here's the nucleus again. Vast majority of cells have a nucleus. But the nerve cell is a very specialized cell that has a long electrical cable-like process that sends electrical signals that can function for sensation or control muscles. Now here is the prion protein. So this is a structure, a model structure of the shape that the amino acids of the prion protein take up when they're folded in the right way. Now the prion protein sits on the surface of a nerve cell, as I've shown there by the arrow, and it's anchored to a membrane that surrounds the nerve cell. So the prion protein has a specific structure, and the main part of that structure is characterized by helices, these shapes, these coils here. Then there are 3, helix 1, 2, and 3, and there are variably occupied sugar sites here. Now, in order to understand how prion diseases work, just imagine that normal shape of the prion protein is this sphere here. And what you imagine is a prion. Now, a prion is a seed-like structure that's formed of prion proteins, but they've all changed shape to a different shape. Now, we don't know exactly what that shape is yet, but we know it's different. And they're all stuck together, and they form a kind of really stable structure that is quite hard to destroy, either with heat or with chemicals. And this structure, the prion, has a property of binding that normal form, and in doing so, it causes it to convert to the abnormal form and therefore grow the seed into a fiber. And that fiber can fragment, making more seeds, which again grow. And so it cycles like this, a chain reaction that makes more and more prions and more abnormal prion protein. Now we think this can be cleared as well, but this process of the prions growing is faster and stronger than the way it's cleared. How do we observe that? These are photographs of microscopic images of brain tissue, and these have been stained by a pathologist with antibodies that show up brown where abnormal prion protein is found. So the consequence of this process for the brain is that abnormal prion protein deposits in brain tissue and causes damage to the brain. All of this in the first place in inherited prion disease is triggered by a mistake in the amino acid sequence of the protein itself. So we do know a lot about the different spelling mistakes that can be seen in the prion protein to cause disease. Some of them we're absolutely confident about, and we've seen lots and lots of patients over 20 or 30 years or so. Some of these spelling mistakes are harmless, do nothing, and some even protect you from prion disease. Some even stop you from getting CJD. And there are some maybe we've just found in one individual ever in the whole world, and we've never seen in anyone else. And for those, we're just not quite so sure yet. And that's one of the reasons to do research, because how do I advise that family? You know, what are the children? Are the children at risk of inheriting that? Is it a perfectly meaningless spelling mistake, or is it one of great significance? So one of the jobs I do is I keep a catalogue of all these spelling mistakes that we've seen and have been seen around the world to help me guide families about what it means to have detected this in a family member, and what the future may hold. So just a little bit more about what that spelling mistake means, okay? So here I've taken one of these chromosomes, one of these coils of DNA that are all wrapped up really tightly. This imagine would be chromosome 20 where the prion protein sits. Now this is at the top here in white. These are the letters of the DNA code that tell the body how to make prion protein, okay? Now I mentioned you need 3 letters for each amino acid. This is the amino acid code. Now, all you need for inherited prion disease is just a single mistake in that code, which I've illustrated in red there, that leads to a single change in the amino acid sequence. And that makes all the difference. Such a seemingly trivial change, but has great consequences. So when you come to families, this would be a typical family tree that we might draw out. And the way we do that is a square is a symbol for a man, a circle for a woman, and a horizontal line means that they had children together. And this is the next generation, then that's the children's generation. A diagonal line means you've died. And if the circle or the square is filled, that means died of the disease of interest. So one thing you can see following through inherited prion disease pedigrees is typically 50% of the next generation are affected. So why is that the case? Well, we each have 2 genes, in fact, for the vast majority of our body's genes. We have twice. We get 1 copy from mum in the egg, and we get 1 copy from dad in sperm. So we each have 2 copies. Now, someone with inherited prion disease has a faulty copy, but just 1 faulty copy. So I've illustrated in this mini pedigree an affected dad who has a faulty gene, but his other gene is fine. And of course, mum has 2 healthy genes. Now, when this couple had children, there's a meeting of a random egg and sperm, right? And that meeting determines the future from the point of view of inherited prion disease, because dad can pass on the sperm either the healthy copy or the faulty copy. So if you look at these 3 children here, this child was unlucky and got sperm that contained that faulty copy, but the other 2 got this healthy copy from dad. Whereas of course, all the children get the healthy copy of the gene from mum. So the 50-50 that we see in pedigrees comes from the fact that we have 2 genes and they're passed on at random. But what does it mean if a baby has inherited a mutation? It doesn't say when the disease is going to start or even if they're going to get the disease, does it? And that that really depends on the detail. It depends on the detail of exactly which error, which spelling mistake is found in the family, because some families have a type of disease that starts quite early in life, and others have a type that starts much later and sometimes never shows up at all. The one I've illustrated here in this chart, it shows the cumulative onset of disease in somebody that carries a mutation for a particular gene mutation called 6-OPRI, which is an extra insert of DNA. And this one can start as early as 20. But some patients that we've seen didn't start until nearly 50. So what the gene mutation determines for an individual depends somewhat on good luck or bad luck. You know, the risk can vary over very significant periods of time for one's life, but it also depends on exactly what the gene mutation is. Coming back to this point, I mentioned that for some mutations that start later in life, those that start typically later in life, we know that some people have carried the gene mutation and haven't got the disease, even though they've lived into a healthy old age. So we know because, say, someone's parent got the disease, and then an individual's child got the disease, but they themselves lived to, say, being a healthy 80-odd-year-old. We know they must have carried the gene mutation and passed it on, but they didn't develop it. And that does happen. It's more common for some mutations than others, and it's very difficult for us to quantify with any precision. But we have got better at doing this and sharing more precise information with certain families that seem to have a low penetrance. Penetrance, the term, meaning the chance that the disease is going to show up in a lifetime. So importantly, for people that have a relative, a blood relative, that has or has died from inherited prion disease, there's the opportunity to have predictive genetic testing. And that's done after genetic counseling. So it's conducted by healthcare professionals who've received special training in the science of human genetics. And the purpose is to make sure that people don't go in to have a test willy-nilly without really thinking about what the consequences might be, both for themselves, for their families, you know, both psychologically, socially, but also financially and legally. And it's part of the counseling process, is part of just doing your duty as a good physician, a good healthcare professional, making sure people are properly informed about what they're going into. But the test is perfect. You know, if you do have the test, there's never a mistake made. This will determine whether you have inherited the gene mutation that caused disease in the family. And of course, once that information is out, it can't be put back in the box. So it's a very important decision. For people that choose to have a test like that, or even those that don't, there's also the possibility of pre-implantation genetic diagnosis. Now this has been taken up on 2 or 3 occasions in the UK, but very rarely. And so I mention this just in case people don't even know what it's about. So this concerns test tube babies essentially. So using this method, a couple, one of whom's either is known to carry a gene mutation or is at risk, choose to make a handful of test tube babies. And those test tube babies can be tested. A single cell can be taken when they're just a ball of cells. No organs are formed or anything like that, just a ball of cells. A single cell can be taken, and that can be tested for the gene mutation. And then only those embryos that don't carry the gene mutation are implanted back in the womb. So mum knows that if that process is successful and she has a baby, that baby won't carry the family mutation. I'm going to just say, before I finish, I'm just going to say a little bit about treatment, because I feel a sense that something's really happening at the moment, and a sense of optimism that things might be different in a few years' time. At least there's going to be some things that are going into rigorous tests. So our own effort here, excuse me, is the development of antibody treatments. So we've been working on an antibody that binds tightly to the normal prion protein and stops it from changing shape. We started to use this treatment in patients with CJD in 2018. We've treated 6 patients. Now, they have all died now, and supplies of the antibody have been exhausted. We're currently analyzing the data and preparing to make a public release of this, but we've certainly been encouraged by the findings so far and are looking to try and take this forward to the next steps. What I want to say is the strategy here is somehow to get rid of the prion protein. That's the problem. Get rid of we know it's likely to be okay to remove the prion protein, whether that is the healthy prion protein or one that contains an amino acid change related to a gene mutation. And there's another very active strategy that's been taken up by an American company in particular to develop a genetic technology that tries to remove the message that the body makes from the nucleus to produce prion protein. And I'm not going to go into any detail about that today, but the proposal this company has manufactured is called antisense oligonucleotide therapy. And that is something the company are hoping to take into clinical trials in the not too distant future. And you know, both of these treatments may well be relevant to people that are at risk of developing disease in the future. In other words, a style of treatment that aims to prevent disease from happening in the first place, rather than to alter it or slow it down once it has started. So I'd just like to acknowledge the rest of the team. Some of them are on this call, I think, although I can't see all the thumbnails. But I think it's a relatively up-to-date photo that includes most of the team from the National Prion Clinic. And this is the Institute of Prion Diseases where I work. And, you know, just to acknowledge all the kind of laboratory scientists that work behind genetic testing, the development of antibody treatments, etc. I'll finish there. Thanks very much.